The neurotransmitter 2, this is the second of the series. The amines, amino acids and peptides. The amine neurotransmitters are catecholamines. They are epinephrine, norepinephrine and dopamine in the brain. They have a single amino group in their structures. The other are serotonin and histamine. They are amines and they also have the amino acid in their structures. The epinephrine, norepinephrine and dopamine are catecholamines and they are derived from the amino acid tyrosine. They are water soluble and 50% bound to plasma proteins in the circulation. The enzyme that degrades them is monoamine oxidase, and the drugs that inhibit the monoamine oxidase are called monoamine oxidase inhibitors or MAO inhibitors. They increase the activity of catecholamines. The production of epinephrine and norepinephrine from the adrenal medulla is a part of flight and flight response. The amino acids. The amino acids are glutamate, aspartate, GABA and glycine. GABA and glycine are the inhibitory neurotransmitters in the brain. GABA is the most abundant inhibitory neurotransmitter in the brain and glycine in the spinal cord. The glutamate and aspartate are excitatory neurotransmitters. The peptides. The peptides are somatostatins, substance P, cocaine and opioids. The catecholamines. The catecholamines are adrenaline, noradrenaline and dopamine. The synthesis and metabolism of catecholamines. Phenylalanine is converted to tyrosine by the action of phenylalanine hydroxylase. This enzyme phenylalanine hydroxylase is present in the liver, and a deficiency of phenylalanine hydroxylase causes PKU, phenylketonuria. The tyrosine then converted to DOPA by the action of tyrosine hydroxylase. This is a rate limiting step in the synthesis of catecholamines. The dopamine and norepinephrine, they act as negative inhibitor or a negative feedback on this enzyme tyrosine hydroxylase. The cofactor for this reaction is tetrahydrobiopterin. The tetrahydrobiopterin acts on tryptophan to convert to 5-hydroxytryptamine also. The pteridine reductase is required for the regeneration of tetrahydrobiopterin. So a deficiency of the three of the above, that is phenylalanine hydroxylase, tetrahydrobiopterin or pteridine reductase will lead to excess of phenylalanine and a decrease in 5-HT. An increase in phenylalanine, that is phenylalaninemia and then phenylketonuria, is a cause of severe mental retardation in children and is a treatable cause. And this is a mandatory test at birth to check for PKU, phenylketonuria. The other mandatory test to be tested at birth is for the cretinism, because these two are the preventable causes of the mental retardation. In PKU, there is a decreased synthesis of catecholamines and 5-hydroxytryptamine. The decrease in a food intake that contains the phenylalanine is the treatment. And the treatment with the drugs is with the L-DOPA that crosses the blood-brain barrier. In the next step of the synthesis of catecholamines, Dopa is converted to dopamine, and then dopamine by under the action of hydroxylase to norepinephrine, and then this norepinephrine is converted to epinephrine under the action of the enzyme PNMT. As discussed earlier, that these two, dopamine and norepinephrine, they act as a negative feedback on the enzyme tyrosine hydroxylase, which is the rate limiting step, and that converts tyrosine to dopa. The norepinephrine that is converted to the epinephrine by the enzyme PNMT, phenylethylamine N-methyl transferase. This enzyme is not present in the nerve endings in the preganglionic fibers, whereas the dopamine hydroxylase is present, so that the norepinephrine is formed, but adrenaline is not formed. Adrenaline is only formed in the adrenal medulla and in the brain, where PNMT enzyme is present. This enzyme is lacking in the nerve endings. The catecholamines are stored in the presynaptic vesicles in a characteristic granulated vesicles. Remember, acetylcholine is stored in the clear vesicles in presynaptic vesicle. But like acetylcholine, they are released by the process of exocytosis. So both acetylcholine and catecholamines are released by the same process of exocytosis. The epinephrine and norepinephrine are formed in the granulated vesicles. Adrenaline, as I earlier told, is not formed in the nerve endings. Only norepinephrine is formed there because of the deficiency of the enzyme PNMT. It's the noradrenaline which is converted to adrenaline under the action of the enzyme PNMT, phenylethylamine N-methyl transferase, present in the adrenal medulla. And this is the granulated vesicle in contrast to the clear vesicles of acetylcholine. So what are the other substances in the granulated vesicle with epinephrine and norepinephrine? The epinephrine and norepinephrine in the granulated vesicles are bound to ADP, chromogranin, which is the binding protein, and the enzyme dopamine hydroxylase. When these substances are released by the process of exocytosis from the vesicles into the synaptic cleft, these substances are also released with epinephrine and norepinephrine, and they come into the circulation. The circulating level of dopamine hydroxylase, which was in the granulated vesicle with them, is a good index of the sympathetic activity, but their half-life is longer than epinephrine and norepinephrine, so it's not accurate. The catabolism of catecholamines. Both epinephrine and norepinephrine are metabolized by two processes: the methylation, number 1, and number 2, oxidation. The methylation is done by the COMT enzyme, catecholamine O-methyl transferase, and oxidation is done by MAO, monoamine oxidase. The COMT, catecholamine O-methyl transferase, converts both adrenaline and noradrenaline to metanephrine and normetanephrine respectively. This enzyme COMT is not present in the nerve endings. It is absent from the nerve endings and is present in the liver and kidney. The norepinephrine is converted by COMT not in the nerve ending, in the liver and kidney, because this enzyme is missing there. The other substance which is absent from the nerve endings, remember, is the enzyme PNMT that converts norepinephrine to epinephrine, and that is present in the adrenal medulla, because adrenaline is formed there. Then methanephrine and norepinephrine, they are converted, both of them, to VMA and MOPAC. The COMT converts both epinephrine and norepinephrine to methanephrine. So in the second step of oxidation, which is done by monoamine oxidase, both methanephrine and norepinephrine are converted to VMA, that is vanillyl mandelic acid, and MOPAC, methyl hydroxyphenyl glycol. As I told, COMT is not found in the nerve endings. Okay, what happens in the nerve endings to norepinephrine, where monoamine oxidase enzyme is present, but the other COMT is absent here. So the MAO converts norepinephrine in the nerve endings to DOMA and DOPAC. DOMA is dihydroxymandelic acid and DOPAC is dihydroxyphenyl glycol. Both DOMA and DOPAC are then acted upon by COMT, that is outside the nerve endings, and are converted into DOMA is converted to VMA, vanillyl mandelic acid, and DOPAC to MOPAC, methoxyhydroxyphenyl glycol. So the final result is the production of VMA from both and MOPAC from both epinephrine and norepinephrine. So where is the MAO found in the neuron nerve endings? The MAO is found in the mitochondria of the nerve endings, and it is also found in the brain, liver and kidney. The COMT is found in liver and kidney but not in the nerve endings. So the measurement of methanephrine and norepinephrine is a good index of epinephrine and norepinephrine secretion. But the excretion is that of vanillyl mandelic acid in the urine. So a urinary excretion of vanillyl mandelic acid for the 24-hour is done to see the activity of catecholamines. Now what happens to the norepinephrine in the nerve endings? See that if there is a reuptake of norepinephrine in the nerve endings here. So the norepinephrine reuptake in the nerve endings, it goes back into the presynaptic vesicle where the MAO is present. So the MAO is not present in the postsynaptic. It is taken up back into the presynaptic vesicle where MAO is present, and this is a major mechanism, reuptake of norepinephrine, by which it is removed from the autonomic endings. So if you do a section of the noradrenergic nerve, there will be more norepinephrine available because of no reuptake. This reuptake will stop, so it will be more available. Now comparison of cholinergic and noradrenergic nerve endings. See, it's not adrenergic, it's noradrenergic nerve endings. So this is a clear vesicle. See that there is the acetylcholine, which is broken by acetylcholine esterase, which is present at the postsynaptic site, and it's broken into acetate and choline, and then choline is taken up back by the presynaptic vesicle here. Not acetylcholine, it's the choline which is reuptaken. But in noradrenergic nerve endings, this is MAO is present here, and it goes to the liver and kidney, and that converts to norepinephrine. Not here, not at the nerve endings, but here at the nerve ending, it is the MAO, monoamine oxidase, which is present in the presynaptic vesicle. So this norepinephrine is actively taken up back into, and then it's converted into DOPA and DOPAC. Okay, so what happens to dopamine? See, dopamine is the first catecholamine to be formed in the synthesis process of catecholamines. In autonomic ganglia and in certain parts of the brain, catecholamine synthesis stops at dopamine. No more adrenaline or noradrenaline. So, and dopamine is also acted upon by MAO, monoamine oxidase, and is converted to DOPAC, DOPAC, dihydroxyphenyl acetic acid. Both dopamine and DOPAC then acted upon by the COMT, catecholamine O-methyltransferase, and then it is converted to HVA, homovalenic acid, and MTA, methoxytyramine. That's what happens to dopamine. The norepinephrine in brain. The norepinephrine in brain is related to anxiety and depression. Increase in anxiety, decrease in depression is related to arousal, and in the cognitive control and the working memory, along with dopamine, with hunger in hypothalamus, respiration, which is the respiratory center in the medulla, two centers, apneustic, inhibitory, and a nematoxic excitatory respiratory center, both in medulla, and the sleep-wake cycle with the serotonin. The norepinephrine is found in locus ceruleus and lateral tegmental field. The lateral tegmental field is in the brainstem, is spinal cord, and olfactory bulb. The norepinephrine in brain parallels with serotonin. The higher concentrations of norepinephrine is found in hypothalamus, and the lower concentrations are found in basal ganglia, because basically basal ganglia are inhibitory. There are two systems of norepinephrine neurons. Number one, locus ceruleus system, and number two, lateral tegmental system. In the locus ceruleus system, there are two types of a descending and an ascending axons. The axon that descends from locus ceruleus to the spinal cord and to the cerebellum, and in the ascending system, the axon, they go to the hypothalamus, the paraventricular and periventricular and the supraoptic nuclei of the hypothalamus, to the thalamus, to the telencephalon, and to the neocortex. So in the locus ceruleus system, the descending goes to the spinal cord, and the ascending fibers they go to the hypothalamus and to the neocortex and thalamus. In the lateral tegmental systems of the norepinephrinic neuron, their cell bodies are in the dorsal motor nucleus of the vagus and the nucleus rectus solitarius at tegmentum. The noradrenergic neuron projects from here, the lateral tegmental system, to the spinal cord, to the brainstem, hypothalamus, and the telencephalon. So these are the two norepinephrine systems. Norepinephrine is concerned with mood. Increased norepinephrine elevates the mood. Decrease in norepinephrine depresses the mood. The MAO inhibitors that interrupt with monoamine oxidase enzyme that degrades the norepinephrine, they elevate the mood. Whereas tricyclic antidepressants, they decrease the reuptake of norepinephrine. So when the reuptake is decreased, there is elevation in the mood. Example of tricyclic antidepressants are desipramine. So the MAO inhibitors inhibit the monoamine oxidase enzymes, and that elevates the mood. Whereas tricyclic antidepressants, they decrease the reuptake of noradrenaline, elevating the mood. And in the cerebellum, the norepinephrine inhibits the Purkinje cell via beta receptors and cyclic AMP. In the anterior pituitary, it inhibits the oxytocin and ADH release. It regulates the pituitary hormones. The median eminence of hypothalamus has noradrenergic and serotonergic nerve endings, and as I told, they both go parallel. Norepinephrine with serotonin is involved in the regulation of body temperature and sleep. How does norepinephrine causes arousal? Both epinephrine and norepinephrine, they produce EEG arousal and behavioral altering by lowering the threshold of reticular neurons in the brain stem. Increase in the blood pressure increases the excitability of the reticular formation, and there are pressure sensitive neurons in the midbrain. The epinephrine in brain. So the neuron that contains PNMT enzyme, phenyl N-methyltransferase, that will convert norepinephrine to epinephrine. And these neurons, they project from medulla oblongata to the thalamus and also to the periaqueductal gray matter and to the spinal cord. So remember, PNMT containing enzyme containing neurons, they will convert norepinephrine to epinephrine, and that happens only in medulla, adrenal medulla, and in the brain, and not in the peripheral nerves. Dopamine in brain as a neurotransmitter. The functions of dopamine is that is the most important, that it plays a critical role in the reward system. It pleasures related to motivation and also to emotional arousal. It's concerned with regulation of motor behavior. It is a low level in the Parkinson disease due to the death of the cells in the basal ganglia. High levels in schizophrenia. Dopamine is concerned with the mood, motivation, motor system function, and reward. The primary mediator for that is the dopamine. So what are the sites of dopamine? Dopamine is found in the SIF cells in the sympathetic ganglia. It is found in the nigrostriatum, the limbic system, neocortex, and in the retina. The dopaminergic system, there are several dopaminergic systems. Number one, it's nigrostriatal system, the project from substantia nigra to striatum. Number two, mesocortical system of dopamine, that project from midbrain to the olfactory tubercle, limbic areas, frontal and the cingulate cortex. Number three, the tuberoinfundibular system, that is intrahypothalamic, the tuberoinfundibular system, and it projects from the cell bodies in arcuate nucleus of the hypothalamus to the median eminence. Number four, tuberohypophyseal system, that connects it to the pituitary, posterior pituitary, from the arcuate nucleus to the posterior pituitary, and that carries oxytocin and vasopressin. The paraventricular system, dopaminergic neuron around third and fourth ventricle. Others, olfactory bulb and retina. Dopamine is involved in pathogenesis of schizophrenia. Amphetamine increases dopamine and norepinephrine and produces psychosis. And also in the pituitary, dopamine is inhibitory to the prolactin secretion from the anterior pituitary gland. The Parkinson disease. The Parkinson disease is an idiopathic degenerative motor disorder of the nervous system, and it may also be caused by head injury, encephalitis, accidental carbon monoxide poisoning, and by pesticide exposure. The pesticide exposure occurs in farmers. Magnesium dust and carbon disulfide exposure with a history of the disease. The drug that causes the Parkinson disease is meperidine. The dopaminergic cell death in the pars compacta and substantia nigra of the substantia nigra and locus coeruleus is the main cause of Parkinson disease. But cell loss also occurs in globus pallidus and putamen. There is also a decrease in norepinephrine in the brain. The pathogenesis of the Parkinson disease. In experimental animals, methylphenyl tetrahydropyridine (MPTP) is converted by MAO monoamine oxidase to methylphenyl pyridine. So MPTP is converted to MPP by MAO, and this MPP, methylphenyl pyridine, is the toxin that inhibits the oxidative phosphorylation and the cell death. Alternatively, endogenous dopamine is quickly oxidized to free radicals, especially if it's in excess and there is increased turnover of the dopamine. The glutathione is also reduced, and the oxidative stress and increase in reactive iron leads to the generation of hydroxyl radical. These free radicals are toxic to neurons, and the oxidative stress leads to dopamine neuron cell death in the nigrostriatal zone. But the sign symptoms of the Parkinson disease do not occur until 80% of the cell die in this region. The disease starts in the middle age or late life and is more common after 65, but the incidence is equal in both sexes. The clinical features are the tremors, resting tremors, rigidity, dyskinesia, and in advanced cases akinesia, and a disturbance of gait and posture. In the tremors, there are rhythmic resting tremors are the flexion and extension of the fingers, hands and toes. And in the forearm, there are rhythmic supination and pronation movement. Number 2, rigidity. There is increased resistance to passive flexion. The cogwheel rigidity, the clasp knife rigidity, these are the features. There is a dyskinesia or akinesia in advanced stage of the disease. There is slowness of the movements in Parkinson disease. The other features are masked facies, slurred speech, shuffling gait, fascinating gait, difficulty in starting and stopping. Handwriting is small and tremulous and illegible, and there is a decreased swing of the arm in walking, and there is blepharoconus and blepharospasm. The Mersenne sign. The Mersenne sign is a repetitive tapping over the glabella produces sustained blink response. There is drooling of the saliva, and there are rapidly alternating movements are impaired in Parkinson disease. The two that remains normal are power remains normal, the muscle power and the reflexes remain normal. The most common psychiatric feature in the Parkinson disease is depression. Dementia also occurs, but occurs in advanced disease. The Lewy body in the neurons. The Lewy body, there are protein inclusions in the neuron, and that protein is alpha-synuclein. The genetic factors in the Parkinson disease is that there is increased incidence in the monozygotic twins, and the disease number 2 is more common in the first degree relatives compared to the controls. The important differential diagnosis of Parkinson disease is done with essential tremors. In the essential tremors, there are movement in the head. It involves the head with nodding or no sign in essential tremors. Whereas in Parkinson disease, it spares the head, but it involves the face and limbs. In Wilson disease, there is early age of onset, the family history, low serum copper, low ceruloplasmin, and acacia-flusher rings in the eyes. The Creutzfeldt-Jakob disease. In Creutzfeldt-Jakob disease, there is a rapidly progressive dementia, ataxia, myoclonus is common, and EEG is characteristic. The Alzheimer's disease. In Alzheimer's disease, there are minor extrapyramidal disturbances with marked cognitive disturbances.